A very big hello, my dear children. Let me see, I am able to see myself. Okay, yes, I am visible. A very big hello, my dear children. So today I am here with the chapter Biotechnology Principles and Processes. So this is one of the chapter which was asked by you guys, which is why I thought I should be doing questions related to this. It's a fun chapter, something very new, probably a fresh one which you have not studied in your any other grades. So biotechnology is fun, it's new, and because of which for some people they feel that this chapter is difficult, but to be honest, it's not so difficult if you know the basics, if you study it properly, if the concepts are really strong, everything becomes easy. And the same thing goes over here, applies for this particular chapter as well. And if at all you think that your concepts are not really strong, I am here to help you out, so please don't worry. I have already made the Brahmastra series where I spoke about all the important questions that come for PU2, and today I am here to discuss the questions that have come in KCET examination in the previous year. So we would be discussing top 20 PYQs. I hope we are ready. Let's go ahead with the first question out there. The only rule that you need to follow is that whenever I show you the question, you can try to solve it by yourself and you can let me know the answers in the live chat session. Okay? I hope that's a deal. So most suitable method of introducing alien DNA into a plant cell is that. So basically there is a gene of interest, something that I need in a plant cell. Probably this gene would be for some pest resistance, such that the plant can live. Plant can survive easily even if the pest is there. So it can provide pest resistance, anything. But anyway, I want to put this alien DNA into the plant cell. What is the method that I would be picking up? Is it biolistics? Is it microinjection? Is it liposuction? Or is it what? Heat shock method. So that is the question. Any guesses? So those of you who are watching this video, do you have any idea what is that method that we use for plant cells? So those who know can comment down the answers. Even if it is wrong answer, please do not mind, my dear children. I am not here to judge you. I am here to teach you, right? So biolistics is where I'll tell you what happens in biolistics. Okay, biolistics is where you use a gene gun and basically you bombard something inside the nucleus directly with the help of some gold and tungsten things. Kumuda is here. Hi dear. Kumuda says the answer for this question is B, that is micro-injection. Is it B? Is there anyone else to say no, the answer is something else? Well, biolistics, as I said, this is the gene gun method. So when do you think a gun can be used? When probably it's very difficult to get inside a cell. And my dear children, this applies for plant cell, because plant cell, my dear children, have an extra wall which is the cell wall, which is why you need a gun, gene gun method. So actually for plant cells, it is biolistics, and I'll tell you why it is not micro-injection. Micro-injection is basically where I have some pipette-like thing and basically I'm injecting the DNA inside the nucleus. This can be done if at all the membrane is not too thick. For example, in animal cells. Okay, so micro-injection is done in animal cells, but when you talk about plant cells, you need gene gun method where you use gold and tungsten thing and you cover it up with DNA and that is being bombarded inside. Liposuction is basically you have your DNA and you cover it some lipid substance and you make it enter. That is not there in our syllabus, so we don't need to worry about that. And heat shock method is usually done in the case of bacteria, where what do? You put a cell first in cold, then you put it in hot temperature. What happens is that all the membrane have temporary pores, and through it whatever DNA we can enter it through that. So that again is not needed here in this particular case, because here you are asked about plants. So your only answer for this question becomes A. I hope it's clear. Is it clear? Kumada, dear, is it sorted? Well, I'm so happy that you tried it out. Super great. Okay, you are doing absolutely great. Now let's go ahead with the next question out there. I think by then children might also come back from school. Okay. So which one of the following statement is not correct about a plasmid is the next question. Which one is not correct about a plasmid? Do you know what is a plasmid? So usually some people might already know it, some people might not know it. Suppose this is a bacteria. Suppose this is E. coli. E. coli will have its own genetic material right around there, which codes for proteins, which is very much responsible for all the functions that is happening in the E. coli. Now E. coli also has some very important thing, which I say it is extra chromosomal. I call this as extra chromosomal content. Why? Because this is not coding for a protein. This is not coding for the proteins which is there in E. coli. It's just there. And this DNA which is there, my dear children, it is very special. You call this as plasmid. It can replicate by itself. Okay, it is extra chromosomal. It might sometimes have genes. It might sometimes have genes that provides the E. coli antibiotic resistance. Antibiotic resistance means, for example, name an antibiotic. Let's take it ampicillin. Okay, and suppose this particular E. coli's plasmid have the gene for ampicillin resistance, then I can grow the E. coli in ampicillin medium also. Why? Because it is resistant to that. So isn't it pretty great? Yes. So you are asked what is not correct about plasmid. Anyone who is watching this video, can you tell me which is not correct about the plasmid right over here? Come on, children. So don't wait for others to answer. If you know the answer, tell me. Listen, this is how we learn. So previous day, the Josh was too much. All of you guys were answering to the question and that was literally fun. But now if at all you're not answering, it would be kind of boring. So please don't do that. Please answer. What is not correct? It is a circular DNA. What do you see? It is a circular DNA. Yes, ma'am, it is. It has antibiotic resistant gene. Yes, it sometimes has. It has the ability of autonomous replication, as in it can replicate by itself. Yes, it has. Why? Because it has origin of replication. A site which is there, very much important for the replication to happen. Now, its DNA is as long as the chromosomal DNA. No, my dear children. It is not. It is not as long as the chromosomal DNA. In the picture it might look like that, but it is not. So your only answer which is wrong would be option D. Okay? I hope that's clear. Now going ahead with the next question out there, I am expecting you guys to answer these questions. So elution, what do you mean by elution is the next question. It means separation of DNA fragments on agarose gel. It means cutting and extraction of DNA bands from the agarose gel. It means making the DNA bands visible under the UV radiation, or it means isolating alien DNA from the choice organism. What do you mean by elution? So elution basically means, suppose I have my DNA fragments and I made them separate using the gel electrophoresis method. Now, once they are separate, I need to take them out also. I cannot keep them forever, right? Keep them there forever. I need to pick them, cut them and take it out. This is known as elution. So over here, separation of DNA fragments on agarose gel, separation of them is not known as elution. Rather, when I cut it and I extract it, I call this as elution. Okay, I will call this as elution. So cutting and extraction of DNA bands from the agarose gel is your answer for elution. This is a very important question. This came in the year 2016. Okay, now we go ahead with question number 4. Children who are watching this video, I am telling you again, make sure you're putting down the answers. It will be fun, it will be nice. Otherwise, the session is going to be super boring. Okay, so restriction endonucleases are isolated from some bacteria. Their role in bacteria is, what is the role of restriction endonucleases in the bacteria? So it is said that bacteria are the one that produces restriction endonucleases. You know what is the meaning of restriction endonucleases? These are enzymes which will be cutting the DNA from the middle, within inside. And it is there in the bacteria and we take it from them. Why do you think they have it? Suppose there is E. coli, there is a bacteria whose name is E. coli. I will call it chutku. Okay, this is chutku. And now this chutku is being infected by a virus, that is bacteriophage. Okay, bacteriophage is infesting what? Infecting chutko out there. So once it infects, my dear children, what does it do to infect? It passes down the genetic material inside. Okay, the virus passes down the genetic material inside the E. coli. Now, if it is passing down its genetic material, it is aiming to get inside or get incorporated into the genetic material of the E. coli. And do you think E. coli will allow it? No. Rather, it will produce this restriction endonucleases and try to cut it off. Now, restriction endonucleases, they anyway cut the DNA. It can cut their own DNA, right? So, to protect it from cutting its own DNA, what does it do? It produces another enzyme which adds methyl group. So two enzymes are there. One adds methyl group and the other one is the one that actually cuts off the genetic material. So the genetic material of this virus is being cut and chutko is now safe. Why? Because it has already added methyl group on itself. So restriction endonucleases are isolated from some bacteria. Their role in the bacteria is to what? Is to provide defense against viruses. They are not as simple as they look. Okay, not for synthesis of protein, not to act as genetic material, not to help in reproduction, but the only answer for this question is to provide defense against the bacteria. This is an important concept. Okay, children, I hope you're understanding. Try to solve question number 5. Come on, those of you who are watching this video, I am pretty sure you're watching it on a purpose. I'm pretty sure you want great marks, right? So we need to try out for the questions. And it is actually becoming to be too boring right here because no one is there to interact. So please make sure, do not be worried about giving wrong answers. Please put down, try to solve it as however you can. So from which bacterium the restriction endonuclease SAL1 is isolated? You have been asked this question, that from which of the bacteria this enzyme SAL1 is isolated. Now, how do you understand? How do you understand that from which bacteria? Ma'am, I have not studied about SAL1 like that. How can I know? So, my dear children, how do you name the enzymes? You name the enzymes according to this one formula, that is S stands for, or the first letter is standing for what? The generic name or the genus name. Mansi is here. Hi Mansi. Mansi says the answer is D. Oh my God. Coming here with a bash. Okay. So this first letter is for the generic name. Two letters. The next two letters are the two letters of your species name. So over here, the generic name starts S. Over here, it is S. But here, AU is there. So obviously, this is not the case. But on the other hand, what Mansi said, it is D. Right, it is D. So over here, S here and ALN here, which is why we can understand that Streptomyces albus is the one from where you can get SAL1. Kudos. Awesome, dear. Okay, now going ahead with the next question, try to solve this. Question number 6. Identify the palindromic sequence in the base sequences. So over here you have given some sequences. You are asked to find the palindromic one. What do you mean by palindromic? The one that you can read same in both the direction. Okay, from 5' to 3' it is read the same, and from 3' to 5' also it is, in both the direction it is read the same, not from 3' to 5'. Any idea? For example, Malayalam. Now, opposite of Malayalam is also Malayalam. Whatever you do, you can read it as Malayalam only, right? So that is a palindromic sequence. And now you have to identify which one is palindromic among them. A, B, C or D. So here 5CGATA is given, but here TA is given. So this is wrong. Let's go ahead with this. GGATTC. GGATTC. You see, it is read in the same way. Mansi says the answer for this is B. I think I also have to lock it as B. Kudos to you, Mansi. Well, over here, my dear children, as you can see, it starts with C, but here it starts with G, so this is not the one. Here also G and C, this is also not the one. The only answer for this question would be B. This came in 2018. Just imagine this easy question comes in your paper. It would be so nice, right? Yes. Now going ahead with the 7th question for the day, the technique of bombarding plant cells with high velocity microparticles of gold or tungsten coated with DNA is. What do you call that technique? Now this is the one that we started off with. This is the question that we started off with. There is a technique where I am going to bombard plant cells. I am going to put gene gun on the plant cells by putting the goalie that I have is actually microparticles of gold and tungsten, and I am coating it with DNA. What do you call this process as? Microinjection, Biolistic method, heat shock method, by disarm pathogen vector. So gene gun is the name of the method, but it has some other name also, which you need to remember. And generally children tend to not think about it. They forget it. But Mansi is not among them. Mansi clearly remembers, which is why she again told me the answer. It is Biolistic method. Always remember, Biolistic or gene gun method, it's the same. So do not be confused. If at all you have to write about Biolistic method. So your answer for this question is B, my dear children. Going ahead with the 8th question. Is there someone to compete with Mansi out here? Seems like there is no one ready to give that challenge, right? Question number 8. A student while extracting DNA from Aspergillus fungus requires dash enzyme to break open the cell wall. What do you think is the answer for this question? A student is trying to extract the DNA, okay? It is he or she is trying to extract the DNA from what? From Aspergillus fungus. It is clearly mentioned it is a fungus. Now, what do you require? Which enzyme do you require to break open the cell wall, my dear children? So, what is the cell wall made up of in the case of a fungus? Cell wall of the fungus is made up of what? Any idea? Cell wall of a fungus is made up of what? In the case of plants, it is made up of cellulose, right? Which is why we need the enzyme cellulase to break it up. In the case of fungus, the cell wall is actually made up of something which is known as chitin, which is why we need the enzyme chitinase. Okay, which is why we need the enzyme chitinase. So always remember that. Hi, Praveen Gowda. My dear Praveen, are we solving the same question? Question number 8, the answer is D. Why? Because cell wall of fungus is made up of chitin, not cellulose. Cellulose is there in the case of plants. And what is, suppose your question was a bacteria is trying to be opened up. In that case, the bacterial cell wall, my dear children, is made up of peptidoglycan. Peptidoglycan. And it is being broken open with the help of lysozyme. In the case of bacteria, it would have been lysozyme. Okay, so 3 things. Plants, cellulase, fungus, chitinase, bacteria, lysozyme. So the tears that we usually have, the tears have a lots of lysozyme, which is why we say tears are actually good. But that does not mean that you have to cry. You guys be happy, okay? So question number 9, identify the DNA sequences which can be cut using EcoRI. So EcoRI is a restriction endonuclease, and you have to tell the DNA sequence which can be cut using EcoRI. The DNA sequence which can be cut using using EcoRI is what? Any guesses by people who are watching this video? EcoRI has a specific site. If at all someone remembers it, you can tell me the answer. What is the site for EcoRI? Any guesses? It is GAATTC, but from 5' to 3'. 5' GAATTC. So which one has that thing? You have to look for that. 5 GAATTC, no. 5 GAATTC, and opposite side also 5 GAATTC. This one has it. But let's go with the other options. Here do you have GAATTC, but that is in 3' to 5' direction. So this is wrong. Here also GAATTC is there, but in other direction. So this is also wrong. The only answer that you would be having would be B. Okay, why? Because EcoRI cuts between G and A of GAATTC. Similarly, here also it will make a cut. Okay, they are palindromic sequences. Next question, question number 10. Come on children, come on. It's like me teaching myself. So you were the ones who said that, ma'am, we want biotechnology principles and processes, which is exactly why I took this chapter. And if I get this in return, I will be literally sad. Okay, identify the labels M and N in the following agarose gel electrophoresis representation. You have to tell me what's M, what is N. What is M and what is N in a gel electrophoresis chamber. So in gel electrophoresis, my dear children, when the DNA fragments are separated, the larger ones stay at back and the smaller ones move fast. The smaller ones are able to move fast, right? So that is the thing that happens. Now I've given you the answer almost. I've actually given you the whole answer. So M is for smallest DNA bands? No. The smallest DNA bands would be there. Okay, M, digested DNA bands. Everything is digested over here. Mansi says 10, the answer is D. Yes, I'm coming to that. Yes, you are correct. M, hybridized DNA bands. There is no hybrid DNA bands over here. So your answer for this question is this. M, larger DNA bands, and my dear children, N is smallest DNA bands which are there. Okay, so this is the answer. This question came in 2020. Going ahead with the next question, question number 11. Suppose DNA samples collected from DNA fingerprinting analysis are less than required quantity. DNA sample which is collected is very less, okay? Which of the following techniques is helpful to make the sample sufficient for the above analysis? I want to do an analysis, but the content is really less. What do I do? Which technique do I have to make a sufficient amount of sample size? I have DNA probing, I have electrophoresis, I have chromatography, I have PCR. Ma'am, this question is super easy. There are two things which are actually not there much in our NCERT and still it is being asked. That means I have to just ignore those things, right? This DNA probing is there but not in exact terms. Electrophoresis means separation of your DNA fragments. What are we left with? PCR. What does PCR stand for? Polymerase chain reaction. Where what do? You basically amplify the amount of DNA. So here when the sample size of the DNA is less, don't you think you're going to use PCR only? Yes, my dear children. So PCR is the technique. And with that I remember one more very important thing. Hi Narasimha, I do not know who is Ammu, but hi. Okay, so anyways, what was I saying is that, yeah, so PCR which is there, it is for amplification, do not forget. So if at all in a question, the question comes like, there is a pathogen in very less amount, then how can you still detect the pathogen? With the help of what technique can you still detect the pathogen? It is again PCR itself. Okay, it will be again PCR. Fine. So because you can amplify then. Question number 12. Try to solve this, my dear children. ROP gene, which codes for the proteins involved in the replication of plasmid PBr322 in E. coli, is located in the restriction site of. ROP gene, my dear children, where is it located? So ROP gene is over here. ORI site of the plasmid is over here. In this site, there is something. This is the site for a restriction enzyme also. You have been asked, what is this restriction enzyme? Is it HIND3? Is it EcoRI? Is it BAMH1? Is it PVU12? PVU2? What is the answer for this question? Mansi says the answer is C. Praveen is there, the answer is D. Okay, so 12, my dear children, the answer is C. It's PVU2 which is there. Okay, restriction enzyme PVU2 is over here. Whereas over here you have PVU1 and also PST1. So two enzymes over here, one enzyme over here, two enzyme over here also, that is BAMH1 and SAL1. So this is the site for ampicillin resistance. This is the site for tetracycline resistance. This is the ROP site. This site means this site is going to help in the production of some proteins which are super important for replication to happen. But restriction enzyme site is also over here for PVU2. Now there are three of the things over here also. One is EcoRI site. EcoRI is there. CLAV1 is there, and also HIND3 site is there. Okay, HIND3 site is there. So basically your answer for this question would be C. Mansi, you are absolutely correct here. And Praveen, we are on the verge of giving our best, right? We are working on it. Chalo. Question number 13. Try to solve this question. What does the sample of a given base sequence represent? So 5-GAA-TTC and again 5-GAA-TTC. What does it represent? Both of them are same. Completion of replication, initiator codon at 5-N, palindromic sequence, deletion, mutation. What does it represent? Both the sequences are super same. It represents what? Palindromic sequence. It represents palindromic sequence. From both the sides it has been read the same, which is why it is palindromic sequence. Okay, see, Alan is over here. Hi, Alan. Okay, Praveen, yes, that's my boy. Kudos. So the answer for this question is C. Now, 14th question. The term molecular scissors refer to what? The term molecular scissors refers to what? Molecular means it is going to cut molecules. Molecules can be anything, DNA, RNA, protein, anything, right? And now you're calling scissors because they cut something, right? So molecular scissors refers to what? Restriction enzyme, polymerase 1, Taq polymerase, polymerase 2. So polymerase means they are going to make something up. So obviously it cannot be this case because you're going to cut something. Everyone is saying A only. Mansi says A, Alan also says A, which is why I'll probably lock the option A. It is restriction enzyme because restriction enzyme cuts the DNA, cuts the RNA, everything. So here in reference to DNA, it is the correct answer. Okay, so you guys are absolutely correct. Now 15th question out there, we just have 5 more questions, my dear children. Which of this is not a method to make host cells competent to take up the DNA? Now this is going to be helpful for you guys. Which one is not a method to make the host cells competent to take up the DNA? That means it has no help in taking up the DNA. This is not a method used for taking up the DNA in simple language. What is the answer for this particular question? Elution, biolistics, use of disarmed pathogen vectors, microinjection. You tell me the answer, I'll explain you other options as well. So, elution basically means what? Elution means extracting or cutting and extracting the DNA from the gel electrophoresis chamber. This is something that has nothing to do with the technique of putting DNA inside. So obviously this one is the odd one out. So yes, Mansi and Alan, you guys are correct, my dear children. Biolistics is a method of putting the DNA inside a plant cell. Use on disarmed pathogen vector is again disarmed pathogen vector. You are again using it for transferring something inside a organism. That is why you call it as a vector. Microinjection is used in the case of an animal cell, where with the help of a micropipette you are injecting the DNA. So 3 of them uses the technique, but this does not, which is why A is your answer. Yes, Praveen, you are also super correct. Try to solve this question. Question number 16. DNA polymerase of Thermus aquaticus is what? DNA polymerase of Thermus aquaticus is what? Thermus aquaticus, my dear children, it's a bacteria. It's a bacteria and it is found at a very high temperature. And this bacteria's DNA polymerase is super important. This bacteria's DNA polymerase is known as Taq, T here and AQ from here. Taq polymerase. Why is it important? Because you know, in PCR method, polymerase chain reaction, you are going to synthesize the DNA at very high temperature using this particular enzyme. So, which means that this particular enzyme has to be, yes, Mansi, yes, Alan, yes, Praveen. The answer for this question would be what? It is B. Who says B? It is D, thermostable. DNA polymerase of Thermus aquaticus is D, not B. D for doll. Okay? Thermostable, it has to be thermostable. High temperature should be withstanding that. Because in PCR, you have very high temperature, right? 95 degrees Celsius, 72 degrees Celsius, things like that. So we need a temperature resistant, something that can be remaining stable at high temperature also. So your answer for this question is D, not B. Okay? Got it? Guys, please correct it, okay? I hope you got it. If at all you got it, just give me a thumbs up. 1 thumbs up would be enough for me to understand that you are not getting things wrong. Yes, thank you so much, Mansi. Okay. Next question, question number 17. Match the content of list 1 with list 2. Thank you, Alan. Okay. So, bioreactors, downstream processing, recombinant protein, PCR is given over here. Always in match the following question, try to match A first and D also. Okay, so A is bioreactors. Insulin produced by RDNA technology, no. Vessels which convert raw materials into specific products, yes, they are bioreactors. Big vessels where our raw material is being converted to what? Our product. Now PCR is polymerase chain reaction, right? Now let's see. Detect mutated genes in suspected cancer patients, involves separation and purification. What do you think is the answer for this question? PCR, detect mutated genes in suspected cancer patient. This is the only thing that matches with PCR. Okay. Nothing else can match with PCR, because if at all you see, recombinant protein, it is insulin. Insulin is something that we can get from recombinant DNA technology. Downstream processing, it involves separation and purification. So your only answer for this question goes like, if at all A goes with 2, A goes with 2 and D goes with 3. A goes with 2 and D goes with 3. I just have one option. Your answer would be this. B goes with 4 and C goes with 1. My dear children, I always tell, try to do this and this first. It will be a lot more easy for you then. Yes, Alan. Yes, Mansi. You guys are super correct. Let's try to solve this question now. Question number 18. The parts of the plasmid that codes for proteins involved in the replication of PBr322 plasmid is. So the part that is involved in the formation of proteins, which are super important for replication is what? So in PBr322, there is a region that codes for proteins, and these proteins are super important for the process of replication. So you are being asked, which is this particular gene or which is this particular part? What do you think is the answer for this question? You have A as ORI site. ORI site is a site that is known as origin of replication. This is from where the replication process will be starting off. Selectable marker is for basically selecting the recombinants or for protecting the cell because they have antibiotic resistance. ROP is something that is ROP. It's ROP. It's not B, it's C, dear. It's C. So you know in PBr322 we have studied this is origin of replication, and just near it you have an ROP site. This ROP site produces proteins which are super important for the replication process to happen. Okay, they are super important for the replication process to happen, and this is where you have the site for the restriction enzyme PVU2. So your answer for this question would be C. Okay, not B. Small things, sometimes we do mistakes, is totally fine. This is how we learn. So do not be sad about it, do not be worried about it, rather just study. Okay, rather just study with me, that's all. Cool? I hope that makes sense, my dear children. Selectable markers are just there. For example, selectable marker, I have a restriction enzyme gene. I have a resistance gene, that is antibiotic resistance, ampicillin resistance. So that means basically this particular organism who has this plasmid will be resistant to ampicillin. So that is a selectable marker. I am able to select the EcoRI that has that resistance. If it has resistance, it means that it has my plasmid also. So your answer for this question is C, not B. 19th question, try to solve it up. To isolate DNA from cells and plant cells, from cells, plant cells and the, okay, the question has some defect in it. So let me see. Okay, let me take from bacterial cells, plant cells and the fungal cells. Now tell me the answer. What is the answer for this question? To isolate DNA from bacterial cells and the plant cells and the fungal cells. What would be the answer for this particular question then? Actually it was mixed, but either way you will get to understand. 19th A, lysozyme, cellulase and chitinase. Yes, Swansea is back in form. And Rukmini, my dear child, I hope you are solving some other question. Okay, just check it out, which question are you solving, because yeah. Okay, so why lysozyme? Because my dear children, as I told you, in bacteria the cell wall which is there, it is made up of something that is known as peptidoglycan, and this peptidoglycan can be broken down with the enzyme lysozyme. Now in the case of plant, In the case of plant, the cell wall again is made up of cellulose. So it can be broken down with the enzyme cellulase. Okay, it can be broken down with an enzyme cellulase. Now the third category is fungi. Ayyo, it went down. Just a second. The third category is fungi. In the case of fungi, the cell wall is made up of what? Chitin. And the enzyme that you need is what? Chitinase. Okay, so your answer for this question, as said by Mansi, is what? A. This is an important question. Please make sure you know these terms. It's super, super important. Okay, now we come down to the last question for the day, and that is this. In the year 1963, the two enzymes responsible for restricting the growth of bacteriophage in E. coli were isolated. One of them cut the DNA and the other added some group. So basically, in the year 1963, two enzymes responsible for restricting the growth of bacteriophage in E. coli were isolated. That means, again I'll tell the story, this is E. coli, and an E. coli was infected by a bacteriophage. What is a bacteriophage, my dear children? Bacteriophage is actually a virus. It is a virus. So when a virus infects it, what it happens is that it basically transfers the genetic material inside. Okay, it transfers the genetic material inside. So over here, you guys are giving me the answer. Rukmini gave me the answer. Yes, and also did Praveen. Yes, you guys are absolutely correct. I'm coming to that. So E. coli has its own genetic material right over here. And what is this virus trying to do? It is trying to get incorporated over here within the E. coli's genetic material, such that its replication and everything can happen smoothly. So E. coli thought that I cannot let the virus live inside my body. So basically, I am going to produce some restriction endonucleases, some restriction enzymes which is going to cleave down the DNA or the genetic material out there. But the problem is that E. coli also has genetic material. It can also be cleaved. The same restriction enzyme can act on itself also. So it had one more enzyme which added one group. What was the name of that particular group, my dear children? It was methyl group. So whoever has the methyl group tag would not be cut by the enzyme. So that was the logic. So in the year 1963, the two enzymes responsible for restricting the growth of bacteriophage in E. coli were isolated. One of them cut the DNA, that was restriction enzyme, and the other one added the group, that is methyl. And you guys were correct by saying that. Kudos to you. And with that, my dear children, we end today's live. I didn't see so many children watching this video, which is why I think that your KCET preparation is not something that you are taking seriously probably, or you might have something else. Is there something happening right now? Is your FA2 going on, or are you preparing more for your PU2? Because if yes, then we are also planning to do something around PU2. So you can let us know and we can help you with that. So if you have any suggestions, as in if you want to tell us anything, you can tell me right now over here. Okay, and please watch the Brahmastra series if at all you are aiming for PU2. It's going to be super good if you practice the questions in Brahmastra. So I assume there is nothing. In that case, my dear children, I'll probably end the live. Wishing you all the best. Thank you so much for those of you who have participated. Lots of love. Please take care of yourself. Okay? Bye-bye.